Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the B cell. In the last video, we talked about the structure of the antibody, and we discussed about the different components of an antibody, like the light in the heavy chains, the constant in the variable regions, the hinge region, and the disulfide bonds bridging the light chain to the heavy chain. Whenever you talk about the antibody, you are going to encounter two more terms. One that is known as the antibody, or this is sometimes also known as the secretory antibody. And the second terminology that you are going to see, that would be the B cell receptor, also known as the B cell antigen receptor. So these are the two common terms you are going to encounter whenever you talk about the B cells or about the antibodies. So let us see about the B cell receptor or the B cell antigen receptor first. Now, this is a journal figure showing you the structure of the B cell receptor. You are going to see the light chain, the heavy chains, the hinge regions, the variable and the constant regions over here. So, whenever you talk about the B cell receptor, you are going to see multiple things. The first thing you are going to see, that is the antigen binding subunit, which is also known as the membrane surface immunoglobulin. And this particular antigen binding subunit is responsible for binding to the antigen. If you look at this terminology, that is going to give you a clue that which type of the antibody you're talking about. And if this is a membrane surface immunoglobulin, so you're still talking about the immunoglobulin, but they are stuck to the surface of the membrane. If you talk about these B cell receptor, how they are then going to be stuck in the membrane of a particular cell, membrane of a B cell. If you talk about this transmembrane domain, this is actually the area that is known as the transmembrane domain of the B cell receptor. And this particular transmembrane domain of the B cell receptor is actually responsible for anchoring this particular immunoglobulin to the surface of the B cell. Therefore, we are calling them as the membrane surface immunoglobulin. Now, if you want to anchor this particular surface immunoglobulin to the plasma membrane of the B cell, we all know that the plasma membrane, the interior of the plasma membrane, is hydrophobic in nature. So what that would mean is that this particular transmembrane domain of the B cell receptor, they should also be hydrophobic in nature. Now, this hydrophobicity, when talk about the living cells, is achieved by multiple mechanisms. You can actually attach the lipid moieties to the proteins once they are made in the process of the post-translational modifications. But in this particular case, you are not going to add any lipid moiety over here. What you are going to do is, if you look at this particular transmembrane domain, again, this is protein in nature, but this is made of the hydrophobic amino acids. So the hydrophobic amino acid, they are going to easily fit into the hydrophobic interior of the plasma membrane. And the common hydrophobic amino acid that you are going to see in the transmembrane domain of the B cell receptor, that would be the leucine, the isoleucine, the valine, phenylalanine, methionine, and the alanine. And of course, there are other hydrophobic amino acid as well, but the one you are going to use when making the transmembrane domain of the B cell receptor, you are going to use these particular hydrophobic amino acids. The second subunit that you are going to see in the B cell antigen receptor or the B cell receptor, that is a signaling unit. And this is actually the signaling unit made up of the Ig alpha, and the Ig beta. And if you look at the Ig alpha and the Ig beta on the interior side of the B cell, you are going to see these immunoreceptor tyrosine based activation motif. We for short call them as the ITAM, and they are very important in the development of the B cell. So whenever whenever the B cell receptor encounters an antigen, this is actually this particular motif, this particular signaling unit of the B cell receptor, which is responsible for the development of the B cell. So if you compare that with the antibody or the secreted antibody, so the differences would be, you are not going to see any signaling subunit. You are not going to see any transmembrane subunit. Therefore, they are actually released into the peripheral blood or into the lymph nodes, for example. So as they do not have the transmembrane domain, they will not be able to get stuck in the interior of the plasma membrane. Therefore, they are secreted. So this is one of the major differences between the B cell receptor and the antibody or the secreted antibody, that in the secreted antibody, as this is not present, so they will not be able to get stuck in the plasma membrane. Therefore, they will be secreted. And the B cell receptor, as they have got this transmembrane domain, they are going to stuck in the plasma membrane. And of course, when we talk about the secreted antibody, there would be no presence of the signaling unit. Now, this is the antibody or the secreted antibody. So you are going to see the light chains, you are going to see the heavy chains, but you are not going to see any transmembrane domain, or you are not going to see any signaling unit. Let us talk about the differences between the B cell receptor and the antibody. If you talk about their location, so the B cell receptor are present on the surface of the B cells. The antibodies, they are secreted into the body fluids like the blood and the lymph. And as I've told you, major reason is the absence of the transmembrane domain in the antibody. Therefore, they are secreted. Another important difference between the two is that when you talk about their role in activation, so the B cell receptor, they are actually activated upon the antigen binding, and this activated B cell would then be responsible for the production of the secreted antibody. So the secreted antibody, they have got no role in the activation of the B cells, and they actually act after the B cell activation. The third important difference between the two is, as I've told you, there is a signaling unit in the B cell receptor. So they are responsible for triggering the intracellular signaling pathway, and the antibodies, they does not trigger any signaling because their main function is in the effector responses, and they are not responsible for the activation of the B cells. If you talk about the antigen recognition, so they are going to bind to specific antigen on the pathogens. That means they are going to bind the whole of the pathogens, and they are attached to the surface of the membrane. When we talk about these antibodies, so they are going to bind to specific antigen in the circulation, for example, in the blood or in the lymph nodes. If you talk about their form, the B cell receptor, they are membrane bound, and the antibodies, they are soluble, and they are the Y-shaped proteins. They are not bound to the surface of the B cells. If you talk about the interaction with the immune cells, so the B cell receptor, they interact with other cells to initiate B cell activation. And when you talk about the antibodies, they are going to engage immune cells, different immune cells like the macrophages and the complement proteins for the pathogen clearance. The Another important difference between the two is that when you talk about the types, so the B cell receptor, they are primarily the IgM or the IgD isotypes in the naive B cells. And when you talk about the secreted antibody, so you are going to see various isotypes. Don't worry about them. We are going to discuss them in detail. But the various isotypes, they are the IgM, G, A, E, and D, depending on the function and the location. And the effector functions, by that I mean that whenever you talk about the neutralization mechanism, so what the B cell receptor do, they are not directly involved in the effector function because they are going to be responsible for the antibodies, the secreted antibodies. And the secreted antibodies are then going to go for different effector functions like the neutralization, the opsonization, and the complement activation. So these are the major differences between the B cell receptor and the antibody. So if I summarize the thing, the transmembrane domain that would be present in the B cell receptor, thereby stucking them into the cell membrane. The secreted antibody does not have any transmembrane domain, therefore they are secreted in the cell. They are secreted in the peripheral blood or the in the lymph nodes. And another important thing is that the signaling unit is going to interact with the B cell receptor, and that is responsible for the activation of the B cells. And once these B cells, they are activated, then they are going to release the secreted antibody. We will continue our discussion in the next video.